Hey guys, we're going to finish off 2.4 proteins today, and you've got some Edpuzzle questions and some notes to do. And I forgot to show you this last time, but you'll notice the first thing on the agenda that I was supposed to do last time was how to live without Miss Sanford. I know, it's tough, right? So before we get into our actual lesson, obviously right now you should be in Canvas and your headphones should be on, otherwise you wouldn't be listening to me right now. If you're an e-learner, make sure you're taking attendance. But instead of going to topic 2, I want you to go to, where do I want you to go? I want you to go to How Do I? Give it a minute and you'll see How Do I Succeed While Miss Sanford's on Leave? Click on that. Okay, so first things first, don't panic. I'll be back. I'll be back before semester exams in January. If we go 2 consecutive weeks with percentages of 5% or less for new cases for COVID, I'll be back as soon as that happens. So if that happens in 2 weeks from now, I'll be back in 2 weeks. So, number one, don't panic. Number two, make sure you stay engaged, right? So just because I'm not there doesn't mean your grades or your exams go away. I'm working at home to make sure that you guys have these lessons and you'll still be able to access them through Canvas just like we've been doing. And I trust that because you're upperclassmen, you will do the work that you need to do because it's your grade, right? I'll do my part, you got to do yours. Now, that doesn't mean that like you're on your own because you're not, right? So I'm still your teacher and I can help you, but you can't necessarily get me a message through Canvas and you probably can't get a message to me through Edsby either. What you can do is you can send me an email here, Sanford Science at 130 at gmail.com. That's like 130, like the room 130. You can always get a hold of me on Remind. That's probably the fastest way to get a hold of me. If you don't know how to sign up for the Remind, in this module, the How Do I, it tells you how to set up for or how to sign on to the Zoom account. So, I'm getting tired. If you have not yet signed up for the Remind, in the How Do I, there's a page that says sign up for Miss Sanford's Remind. Okay. I'm also going to be holding office hours on Zoom. So that means Tuesdays and Thursdays from 6 to 8, shoot me a remind and I'll meet you on Zoom and we can talk about whatever you need to. Here's the link and there's the password. Same thing Sunday, 6 to 9 p.m. or by appointment. So say you got something to do Sunday night and you want it to be during the day. That's fine. Or say Tuesday, Thursday, or Sunday doesn't work for you. It's got to be a Monday or a Wednesday or a Friday. That's fine too. Just shoot me a remind and I am happy to meet you online. Okay. And we can talk about whatever it is you need to talk about. I can walk you through lessons, I can walk you through questions. Um, so I'm still your teacher. I can still help you, but you've got to reach out and let me know how I can help. The other thing that's really going to help is make sure that you're submitting work on time. So I'm not going to be able to access Canvas like every day to unlock something or reset it for you. If I'm lucky, I'm going to get like access once a week. So the best thing that you can do to help me out is make sure that your work is in on time so that when I do get that access, I can get everything graded and get it done so that you get grades in a timely fashion. And last but not least, just be the responsible and talented student that you are. I mean, you're an IB student for a reason, and that means something, right? So, in the IB world, we step up to challenges that are presented to us and we do what it takes to make things work. And I'll be doing my best to make sure that you have what you need while I'm gone. And your job is to take advantage of it and do your best because that's all you can do. Okay, so let's go back and actually continue with our lesson today. So, last time we talked about proteins. We reviewed topic 2.1, skill 1, molecular diagrams of generalized amino acids and their structure. We talked about how amino acids are linked together by condensation reactions to form polypeptides. We talked about how to draw molecules to show the formation of that peptide bond. We talked about how there's 20 different amino acids in polypeptides and they're synthesized by ribosomes. We talked about how they can be linked together in any sequence, giving a huge range of polypeptides, right? So there's 20 different amino acids. When you multiply 20 factorial by the number of amino acids that are in a protein, you get millions and millions and millions of options. And we also talked about how the amino acid sequence of polypeptides is coded for by your genes, right? And we all speak the same genetic language. That's why you can take one gene out of an organism and put it in another, and then that organism will make the protein of the first. And probably the classic example of that is there are fish that swim in the Arctic, and they are really good at maintaining life in super cold waters, like almost freezing. And they do that because they produce a particular kind of protein that keeps them from freezing. So scientists have isolated that gene and they've replicated that gene and put it in certain farm crops like tomatoes. So now, if there's a freeze while those tomatoes are growing, those tomatoes don't freeze. And it's because the tomato knows how to make that protein because we all use the same 20 amino acids and we all use the same base pairs for genes, A, G, T, C. So now we're going to pick it up and we're going to continue with understanding number 5. After being synthesized, proteins can consist of one or more polypeptide chains. Each chain is a long sequence of amino acids, and in your notes, we're at understanding number 5. A protein may consist of a single polypeptide or more than one polypeptide linked together. So here we have a long string of amino acids, right? And if you were to look at the molecular structure, it would look something like that. We have our hydrogen, we have our R group, we have our central carbon, we have our amino group, and we have our carboxyl group. So there's our ribosome. There's an amino acid. Our ribosome moves over and attaches another amino acid. It moves over and attaches another amino acid. So there we go. All right, looks good. Okay, so here are some examples of polypeptides. There's lysosome, which is an enzyme that digests peptidoglycan in cell walls. So a lysosome, lyso means to break. So if you're lysing something, you're breaking it. So this is something that's really good at breaking down cell walls. In fact, there are some antibiotics that use lysosomes to help break down the cell walls and bacteria that are trying to make you sick, right? And this is actually what it looks like. The second one, and this is one you definitely should know, we're going to refer to it often throughout the next 2 years. This is hemoglobin, and this is used to transport oxygen through the blood. And you'll notice that it's got 4 polypeptide chains. It's got one here, it's got one here, it's got one here, and it's got one here. So I'm going to give you a minute to get all that written down. Okay, so hopefully you've got that down. If not, don't worry. This video is available. You can come back and look at it again, okay? Understanding number 6. The amino acid sequence determines the 3D or the three-dimensional conformation. Don't be afraid of the word conformation, just means shape of a protein, right? So the sequence of amino acids or the order, the primary structure of the amino acids is determined, right, by the order of amino acids. Did I just say that right? It's late. I put in like 13 hours today. Let me try that again. Okay. The amino acid sequence determines the 3D shape of a protein, right? Okay. So a protein's shape is determined by the order of amino acids. Remember, your form equals your function. So, amino acids in a polypeptide chain interact with one another. Remember, sometimes they're charged or sometimes they're polar, and that causes them to bend in weird, funky ways. And it's those interactions between those R groups that determine the three-dimensional shape of a protein. So, here you can see this is just a chain of amino acids. This is what we would call the primary structure, or the order of amino acids is the primary structure. And then secondarily, we have whether they form bent sheets. These are called pleated sheets. And this is the secondary structure, right? Or you can have a helix like DNA, but just one, right? So this is your, how about this? We'll write up here. This is your primary structure, and that's basically just the order, right? This is your secondary structure, and that's whether it forms a pleated sheet like a piece of paper bent into a fan, or whether it's a helix like a curlicue like DNA. And then you have tertiary structure, and that's how those two things come together. So you've got kind of this weird bendy pipe cleaner looking thing now. That's tertiary structure. So tertiary structure is how primary and secondary structure interact together to form a three-dimensional bendy pipe cleaner thing. And then, oh, look at that. See, I have it right here. Primary structure, secondary structure. Now this can be a pleated sheet like a fan, or it can be a helix. So helix, right, or a fan. And then there's your tertiary structure. And that's when you get the whole thing bent up like that. We talked about this freshman year. I did the arm thing where you stick your arms out, primary structure, and then secondary structure was like your elbows bent, and then tertiary structure was one arm in front of you and one arm behind you. And I'm doing it right now, but you can't see it. And then here you go. Here was more of that primary structure, just the order of the amino acids. Then you've got your secondary structure, and it's a pleated sheet. It's bent like a fan. And there are other little hydrogen bonds that happen. Right, so it's not just 1, 2, 3, 4. Now you've got these extra bonds. You can form a sheet. And then finally, you have what's called quaternary structure. Quaternary means fourth, right? And that's when you have multiple peptide chains together. So there's 4 here. And this is really, now we have a for real protein. So primary, secondary, tertiary structure, those are all polypeptides. You're not a really like grown up protein until you've got multiple polypeptides together, and that's a protein. And we have 4 here. So let's see if I can. Here's 1. And then here's 2. Where does that go? I think it's back here. And then we have 3. That one's pretty obvious. And then we have 4. And you can't see any of the ends of that one, but one's back here. Don't worry, I'm not going to like make you untangle all that spaghetti monster mess. What you need to know is primary structure, secondary structure, tertiary structure, and quaternary structure. And by the time you get to quaternary, that's multiple polypeptide chains together. That's when you have a protein. Okay, application number 2, denaturation of proteins by heat or by deprivation of pH from the optimum. Okay, so under normal conditions, the three-dimensional shape of the protein is stabilized by bonds between the amino acids, right? So here we come back to those all important hydrogen bonds. All important hydrogen bonds, your side chick bonds, right? So you've got this oxygen that's making eyes at this hydrogen. They're on 2 different amino acids and they're going to form that kind of weak bond. Now, this is not a polypeptide bond, right? This is not a peptide bond between 2 amino acids. This is a hydrogen bond between different numbered amino acids. So, this could be amino acid number 1 over here, and here's 2, and here's 3, and here's 4, right? So you get hydrogen bonds between different amino acids, and that helps maintain that shape. But because these are just side chick bonds, they're not like for real, different environmental conditions can actually break those bonds. So denaturation occurs when a protein loses its structure or loses its shape, and that can be because temperatures are too high, like when you have a fever. A fever is literally your body heating itself up because it's trying to break the proteins of whatever is invading your body. So fevers, they're a defense mechanism. Your body is heating up to actually break the proteins of whoever is trying to make you sick. Changes in pH can do the same thing. So if a particular protein is designed to function in acidic conditions and all of a sudden it's in basic conditions, it's going to lose its shape and vice versa. And salinity can actually do it too. So if salt concentration is too high, that can also break a protein. So here we see on the left, we see a protein with all its nice curly cues. It's got all that primary structure and secondary and tertiary and quaternary. There's no quaternary there, but there's tertiary structure. Something happened. This protein either experienced temperatures that were too high or pH conditions that weren't great or changes in salinity that caused it to lose that shape. Now, you can imagine if this protein needed all these curlicues to do a very specific job, clearly it's not going to be able to do that job anymore if it's nice and straight. I shouldn't say nice and straight because it's not nice. Clearly it can't do its job if it's been bent. So think about slinkies. Like when you bend that slinky, it never goes back to the way it was and it's never quite right. It never functions exactly like it should. So denaturation, it's like bending a slinky. You're going to break that shape. Here we go. Here's a neat little video that kind of just says the same thing that I did. So let's see.
Extreme temperature, pH, and salt concentration can change or destroy the structure of a protein, and consequently change or destroy its activity. An egg provides a good example of how heat can change the structure of proteins. Proteins are found in both the egg white and yolk, and these proteins have different structures in raw versus cooked eggs. Let's consider the proteins in an egg white. In their native uncooked state, the proteins are chains of amino acids that fold into complex shapes. Several types of bonds form between amino acids in a chain, stabilizing the structure of the protein. When the egg is cooked, the heat makes the molecules vibrate. The bonds linking the amino acids in chains are strong covalent bonds that withstand the heat. However, most of the bonds that stabilize the three-dimensional structure of the protein are weak bonds which break more easily, allowing the proteins to unravel. The destruction of a protein's three-dimensional structure is known as denaturation. When two unfolded proteins make contact, they form new bonds with each other. A large net of interconnected proteins forms, and the egg hardens. Light can no longer penetrate the egg, which is why the clear portion of the egg turns white.
All right. So egg whites are basically denatured proteins. When the proteins are maintaining their original shape, the egg white is clear, and then as it heats up, those proteins change their shape. They form a network and bond with other kind of stretched out proteins, and it turns the egg white solid, and you can't see through it anymore. So, understanding number 7. Living organisms synthesize lots of different proteins with a wide range of functions. And here are several that you have to know. So, blood clotting. You couldn't clot blood without proteins. And that's proteins that are found in your blood plasma. We'll talk more about that next year when we talk about the blood system. Catalyzers, right? So, enzymes are a huge class of proteins that we're going to talk about next topic, and they are responsible for the majority of the metabolism, the anabolic and the catabolic reactions that happen in your body. So, really, like life is just a bag full of chemical reactions, and without enzymes, none of that can happen. So, you really kind of owe proteins your life, like literally. Next is membrane transport, and we'll talk about that when we talk about topic 1.3 and 4. You may remember diffusion from when you were a freshman, or active transport, endocytosis, and exocytosis. Those are membrane proteins that help move stuff across membranes. There's tensile strength. We talked about that a little bit last time when we talked about collagen, and it provides strength to skin and ligaments, blood vessel walls too. And then immunity, that's a big one, right? So proteins actually can produce antibodies that allow them to identify and attack pathogens. So proteins basically are some proteins are like special ops. They produce antibodies that go tag targets. And then there's cell adhesion. That's what allows cells to stick to one another, kind of important so that you don't like literally disintegrate. And then there's DNA packaging too. So when we talk in topic 2, the end of topic 2 and topic 3, we'll talk about how eukaryotes have DNA that's coiled into chromosomes, and there's actually little protein balls in the middle of those chromosomes that hold the DNA together. And we'll talk more about that later. So you can see these are all things that are pretty important. You use pretty much all these things on a daily basis. So proteins, lots of different functions, and that's because they have so many different shapes. And that's because we have 20 different R groups and we can put them in a million different orientations. So proteins, super diverse, super functional, super important. Two specific enzymes that you should know. One is Rubisco, and it catalyzes the reaction that fixes carbon dioxide from the atmosphere. So, what does that mean? So, okay, you think of a tree, right? And trees use photosynthesis to produce glucose, right? Well, if you think about the mass, like the whole mass of the tree, that mass is carbon based, and they get that carbon from carbon dioxide. So, plants are awesome because they literally turn gas into solid matter. So CO2 from the atmosphere comes in during photosynthesis, and that carbon stays behind, and it's actually what builds the mass of the tree. So that's what Rubisco does. It's an enzyme that helps that process of converting that CO2 gas in photosynthesis into mass. So a tree is basically like solidified carbon from the atmosphere, which is why it's such a big deal when we cut all the trees down, because then we're literally taking the things away that help us suck carbon dioxide out of the atmosphere, which we kind of need right now. The next protein that you need to know is insulin. And anybody that is diabetic or has a family member that's diabetic knows all about insulin. So, insulin is actually a hormone, and it helps control blood glucose levels in the body. It tells your liver and muscle and fat tissues to take in glucose that's in your bloodstream. If you are type 1 diabetic, that's usually you get type 1 diabetes in your childhood because usually your immune system goes like haywire and it attacks your own pancreas. And that's a big thing, right? So your body no longer has the ability to produce insulin. It's your pancreas that produces insulin. So you have to take insulin instead. Type 2 diabetes is different. Type 2 diabetes is when you eat sugary foods so much that your body basically ignores insulin. It doesn't mean anything anymore. Like, when your parents nag you to do something over and over and over again, and like eventually you basically don't even hear them anymore. That's kind of what type 2 diabetes is. It's insulin is flowing through your body. You have enough insulin, but your body's not listening because it's just used to having a lot of sugar all the time. But insulin is a hormone, and this particular hormone is a protein. So proteins are really very diverse. They are chemical messengers in the body. Here are the immune proteins that you need to know. They're called immunoglobulins. They kind of sound like goblins, but these guys are basically tags. So if you play any kind of like combat games or you know anything about the military, before a major strike, you usually have like a special ops group that goes in and they laser tag buildings before a hit, and then you'll have like air support that'll come in and drop missiles, and the missiles hone in on those laser tags. Well, your immune system is actually like really similar, right? So your immune system has immunoglobulins. They're like your special ops team. They go in and they produce antibodies that only fit to specific uh pathogens or invaders in your body. So these guys, they tag. So here's like an invader. Here's a bad guy, right? And these immunoglobulins, they go in, they produce these antibodies, and the antibodies tag the pathogen so that later these big macrophages, are like basically giant Pac-Men, come by and they eat this and destroy it. And the only reason they can see it is because it's got this like laser tag on it. We have like a whole subtopic on the immune system that we'll do next year. It's really fun. Your immune system like literally is tactical warfare. Like you can make analogies with Star Wars, you can make analogies with all sorts of like first person shooter games. There's all different positions that you can play and it's all like it's amazing how much human war mimics what's going on in your own body. It's nuts. Anyway, okay, let's talk about rhodopsin. Rhodopsin is another protein that you need to know, and this is a membrane protein, meaning it's found in a cell membrane in the retinal rod in your eye. So your eye, it actually looks kind of like a grape, right? And this is like where your eyelashes are. So this is the front, and this is the back of your eye, like in your head. So these guys are back here, right? That's where the cells are. And they absorb light and initiate nerve impulses to your brain. So this is where they are. And these guys allow light to pass through, which triggers a nerve cell, and that actually sends stuff to your brain so that you can actually see. So that's pretty important. Proteins that let you see. Next is collagen, and we talked about collagen already. Collagen is the rope-like protein. You can find it in your skin, and it provides structure and structural support. It lets your skin bounce back so you don't look like a dented banana. And then spider silk. Like, I am not a big fan of spiders, but let me tell you, spider silk is like phenom. We are learning how to replicate different kinds of spider silk to make body armor that's lighter and stronger than Kevlar. And you got to think, like, spiders have been around for a long time. They've been around for millions and millions and millions of years. And when it comes to producing fabrics that are like way better than anything humans can produce, they are it. It's phenomenal. Like down to the molecular level, they have it pat. They know exactly what kind of threads to build to do different functions. And all those threads are all built by different kinds of proteins, right? So spider silk is used by spiders to form the spokes in the webs. They also make drag lines with them. Um, they have there's actually a really good TED talk that we'll listen to about this. And like I said, stronger than Kevlar. Like this stuff's no joke. And you kind of like if you've ever walked through a spider web and you feel it on your skin or on your face and you feel like you can't ever get it off, it's because that stuff like literally doesn't break. Some of it does. The weaker these threads will, but the actual spokes, these are like darn near unbreakable. There's a little tiny spider making something that humans can't even do. It's crazy. Okay, so that was a bunch of different examples of different kinds of proteins to help give you an idea of the diversity of function and form and different types of life that actually produce different kinds of proteins for different things. So, understanding number 8, every individual has a unique proteome. Proteome. So, if a genome is all the genes that exist in a species, the proteome are all the proteins that are produced by a specific species or a specific individual. And we use a process called gel electrophoresis to identify different types of proteins. And all that means is that we put them in a plate and we charge them. And as they carry a charge, they move different distances based on their weight and size. And so we get kind of a spread. So here is a sample of like, I don't know, an extract of something with proteins. We separate it and then we put it in what's called a gel. You take a pipette. Actually, I have a video of me doing this. Maybe I'll find it. And then we charge it, we run an electrical current through it, and it's this way. And then the proteins actually move through a gel, and the further they move, the lighter they are. which makes sense because if you're really heavy, you can't go very far. If you're light, you can travel faster or further. And we use this to figure out by the molecular weight and size all the different types of proteins that are produced by an organism. So, an organism's proteome is the library of proteins that it is able to synthesize, and it's unique to every individual and to every type of cell. So, lots and lots of proteins that can do lots and lots of things. All right, so here's some review. Outline the formation of a dipeptide from two amino acids. I think you did that last time. Name the organelle responsible for protein synthesis. What molecules encode the sequence of amino acids? Outline the role of amino acids in a protein's 3D conformation. And five, describe protein denaturation and what can cause it. So take a few minutes and you can answer the questions provided for you. And then there's a TED talk that you can watch, and I'll ask you some questions about that. And that concludes the notes for topic 2.4 proteins. Remember, when you're done with this video, go back, fill in your notes, look through the 2.4 skills, applications, make sure you have everything that you need. Okay. All right. Talk to you guys next time.